Hello everyone, welcome back to CAPSA. In today's video, we are going to discuss acetylcholine, one of the most important neurotransmitters in the human body. Stay with me till the end of the video because we will cover everything: its chemistry, synthesis, receptors, functions, and even clinical relevance. So first, what exactly is acetylcholine? Acetylcholine, often abbreviated as ACH, is a chemical messenger that allows nerve cells to communicate with other nerve cells, muscles, and glands. It was the very first neurotransmitter to be discovered way back in 1921 by Otto Loewi, who called it Vegastov. A neurotransmitter is a chemical that transmits signals across a synapse, the gap between two neurons or between a neuron and a muscle cell. Chemically, acetylcholine is an ester of acetic acid and choline. You can imagine it as a small water-loving molecule. It is quaternary ammonium compound, and meaning it carries a permanent positive charge, and because of it, it cannot cross lipid membranes easily. This is why it does not cross blood-brain barrier significantly. Now, how is acetylcholine made? The process takes place inside cholinergic neurons, that is, neurons which release acetylcholine. The enzyme which is responsible is called choline acetyltransferase, often shortened as CHAT. This enzyme combines choline, a nutrient obtained from our diet and recycled from previous acetylcholine breakdown, with acetyl-CoA, which is a molecule generated inside the mitochondria during energy metabolism. The result of this reaction is acetylcholine plus coenzyme A. Once synthesized, this acetylcholine is not left free in the cell. It is stored safely in a teeny pocket called synaptic vesicles. The transporter that loads acetylcholine into these vesicles is called VACHT, or vesicular acetylcholine transporter. The vesicular acetylcholine transporter is a transporter that can store the acetylcholine. So when a nerve impulse or action potential reaches the end of the nerve terminals, it causes calcium ions or calcium channels to open. Calcium ions rush in, and this triggers the vesicle to fuse with the nerve membrane, releasing the acetylcholine into the synaptic cleft. This process is known as exocytosis, and importantly, it is calcium dependent. Now let us see how the action of acetylcholine is terminated. Unlike another neurotransmitter such as norepinephrine, acetylcholine is not reuptaken intact into the presynaptic neuron. Instead, its breakdown happens right in the synaptic cleft very rapidly. This process is carried out by an enzyme called acetylcholinesterase, often abbreviated as ACHE. This enzyme splits acetylcholine into two products, choline and acetate. Here, acetate simply diffuses away, but the choline part is very important. Choline is taken back into the presynaptic neuron through a specialized high affinity transporter, where it is again used for the resynthesis of fresh acetylcholine. This recycling process ensures a continuous supply of acetylcholine for the next nerve signals, while at the same time keeping the neurotransmitter's action short and tightly controlled. Now comes the important part: how does acetylcholine act? So it binds to two broad classes of receptors: nicotinic receptors and muscarinic receptors. Nicotinic receptors are ionotropic receptors, meaning they are ligand gated ion channels. When acetylcholine binds, these channels open to allow sodium and potassium ions to move causing depolarization and excitation of the target cell. Nicotinic receptors are present at the neuromuscular junction of the skeletal muscle and in the autonomic ganglia. On the other hand, muscarinic receptors are metabotropic receptors, which means they are linked to G proteins. They do not directly open ion channels but instead trigger signaling cascades inside the cell. There are five types named M1 to M5. For example, M2 receptors are found in the heart and slow down the heart rate, while M3 receptors cause smooth muscle contraction and stimulate the glandular secretions. Where do we find the acetylcholine in the nervous system? So, in the peripheral nervous system, it is released by all preganglionic neurons of the both sympathetic and parasympathetic divisions, and also by all parasympathetic postganglionic neurons. Keep in mind, these are very important for your exams. It is the neurotransmitter at the neuromuscular junction, and interestingly, even synaptic postganglionic neurons that innervate the sweat glands release the acetylcholine. In the central nervous system, acetylcholine is present in the basal forebrain and brainstem, where it plays a major roles in learning, memory, attention, and arousal. Now let us talk about the physiological roles of acetylcholine. At the end, at the neuromuscular junction, acetylcholine is the key neurotransmitter that stimulates the skeletal muscle contraction. Whenever a motor neuron fires, acetylcholine is released onto the muscle end plate, leading to depolarization and contraction of the muscle fiber. Moving on to the autonomic nervous system, acetylcholine plays different roles in the parasympathetic and sympathetic divisions. In the parasympathetic system, it causes bradycardia, meaning it slows down the heart rate. It produces bronchoconstriction in the airways, increasing glandular secretion such as salivary and gastric secretion, and enhances gastrointestinal motility, which means it promotes the digestion. In the sympathetic nervous system, acetylcholine is also released, but in a very specific place by postganglionic fibers that supply sweat glands. This is why sweating is a cholinergic function, even though it belongs to sympathetic division. In the central nervous system, acetylcholine is heavily involved in the higher brain functions. It plays a vital role in memory, learning, attention, arousal, and reward pathways. A deficiency of cholinergic activity in the brain is strongly linked to disorders like Alzheimer's disease. Finally, looking at the cardiovascular system, acetylcholine decreases heart rate and slows conduction of the impulse through the heart by acting on the M2 muscarinic receptors. These parasympathetic effects is essential for maintaining balance with sympathetic stimulations. When we focus on the central nervous system, acetylcholine plays an especially important role in the higher brain functions. It is heavily involved in attention, learning, and memory. In simple words, it helps the brain stay alert, process new information, and store that information for later use. A deficiency of acetylcholine in certain brain regions, particularly in the basal forebrain, is strongly linked to Alzheimer's disease, which is a neurodegenerative disorder characterized by progressively memory loss, cognitive decline. That is why drugs that enhance cholinergic transmission, especially acetylcholinesterase inhibitors like donepezil and rivastigmine, are commonly used in the management of dementia. These drugs prevents the breakdown of acetylcholine, allowing more of it to remain active in the synaptic cleft, and in this way they temporarily improve the memory and cognitive functions in the patients. Now you might be wondering, if acetylcholine is such an important neurotransmitter, can we use it directly as a drug? The answer is no. Acetylcholine itself is not used clinically because of two major reasons. First, it is rapidly broken down by acetylcholinesterase. So, if we gave it as a medicine, it would be destroyed almost immediately and have no sustained effect. Second, it is non selective, meaning it activates both muscarinic and nicotinic receptors all over the body. This would lead to widespread, unpredictable actions, making it unsafe and impractical as a drug. Instead, what we do in medicine in use of cholinomimetic drugs, which are synthetic agents that mimic the acetylcholine activity but are more stable and selective. Examples include bithanicol, carbachol, and pilocarpine. Another approach is to use anticholinesterase drugs like the neostigmine or physostigmine, which prevent the breakdown of acetylcholine and thereby increases the level of its level at the synapse. Now let's look at some important clinical conditions related to the acetylcholine. So the first one is Myasthenia gravis. This is an autoimmune disorders where the body produces antibodies that destroy the nicotinic receptors at the neuromuscular junction. Because of this, acetylcholine released from the nerve cannot effectively stimulate the muscle, leading to weakness and fatigue. The treatment involved acetylcholinesterase inhibitors such as neostigmine, which increase the amount of acetylcholine available and improve the muscle strength. Next is Alzheimer's disease. In this condition, there is a loss of cholinergic neurons in the brain, especially in the areas important for memory and learning. This reduction in acetylcholine contributes to progressive memory loss. Drugs like donepezil, which are acetylcholinesterase inhibitors, are used to slow down the symptoms by maintaining higher acetylcholine levels in the brain. In Parkinson's disease, there is an imbalance between dopamine and acetylcholine in the basal ganglia, the part of the brain that controls movement. Too much cholinergic activity compared to dopamine leads to tremor and rigidity. This is why anticholinergic drugs are sometimes used as part of the treatment. Another example is botulinum toxin poisoning. So, botulinum toxin blocks the release of acetylcholine from the nerve endings, leading to paralysis of the muscle. This is a life threatening condition, although the same toxin is used in very small controlled doses in medicines and cosmetology. Finally, we have organophosphate poisoning. Organophosphates are chemicals found in some insecticides and nerve gases. They irreversibly inhibit the acetylcholinesterase enzyme, which causes an excessive buildup of acetylcholine. This results in overstimulation of both muscarinic and nicotinic receptors, producing what is called a cholinergic crisis with symptoms like salivation, sweating, muscle twitching, and potentially the respiratory failure. Now let's quickly review some important drugs and toxins that acts on the acetylcholine system. So cholinergic agonists like bithanicol, pilocarpine mimic the acetylcholine and directly stimulates its receptors. Cholinesterase inhibitors such as neostigmine, physostigmine, and even organophosphates increase the acetylcholine levels by preventing its breakdown. On the other hand, anti-muscarinic drugs like atropine and scopolamine block muscarinic receptors, while nicotinic antagonists such as teiboquirine and hexamethonium block the nicotinic receptors. Finally, we have toxins. Botulinum toxins prevents the release of acetylcholine leading to paralysis, whereas the black widow spider toxin causes an excessive release of acetylcholine producing severe muscle spasm. Finally, a quick word on acetylcholine as a drug. Pure acetylcholine is not used clinically because it is broken down very rapidly and acts on multiple receptor types, making it non-selective. Instead, synthetic analogs or drugs that affect its metabolism are used in medicine. So that was a complete discussion on acetylcholine, covering its chemistry, synthesis, receptors, physiological roles, and clinical importance. I hope this lecture helped you build a strong foundation on this vital neurotransmitter. Don't forget to like, share, and subscribe to the channel and stay tuned for more lectures in pharmacology, physiology, immunology, microbiology, this kind of subjects.